hear me all right?
Yes, we can hear you.
Perfect. Okay. Can you see my screen?
Yes, it works perfectly.
Okay. Wonderful. Thank you very much for the invitation. It's a real pleasure to contribute to this event. And like my colleague, I'd like to start by thanking the committee for giving us this great opportunity to discuss our work. So I really want to understand how plants develop, evolve, and respond to change. And in particular, one aspect I found absolutely fascinating is how decision made by cell at the microscopic scale can have impact at the macro scale. For instance, affecting the way tissue are patterned, organs are shaped, and even how plant can interact with other living organisms or respond to the environment. And petal pattern really are a fantastic system to study this phenomenon, because the decision made by cell at the microscopic scale are readily observable on the petal surface. And also because these petal patterns fulfill all sorts of fantastic functions. They are involved, of course, as we've seen in the previous talk, in plant-pollinator interaction, either before landing or after landing, to guide the pollinator to the reward. These patterns are not always visible to us. Many are in the UV range of the spectrum, but they are visible to the animal that pollinate the flower. And some really interesting recent work have shown that some of these patterns might represent some adaptation to environmental factors, such as providing pollen protection against UV radiation, for instance, or even participating in reducing water loss in dry environment. So this pattern can be really intricate, like the one we see on this beautiful orchid flower, except this is not an orchid. This is an orchid mantis. And this insect is mimicking the pattern and the shape of the flower to trick pollinator and lure them to their death. So I don't know if this quote applies to human, but it definitely applies to the interaction between plants and the insect that pollinate them. So petal pattern, they come in all sorts of shape and form, but they always involve some form of regulation of the local production of pigment. But they do more than that. They also rely on the control of cell shape and texture, cuticle texture, of the petal surface, because more often than not, we find that region of the petal that produce different color also produce different cell shape covered with different texture. And these structural elements, cell shape and cuticle ornamentation, shouldn't be surprised that they often correlate with region with different color, because these features can directly impact on the appearance of the flower by playing with the light. For example, this is a daisy that grows in the Cambridge Botanic Garden, and it appears really shiny blue at the base, but there is no blue pigment in this flower. And the color is simply produced by, it's an optical trick, because the light is diffracted by microscopic striation that cover the surface of the epidermis cell in this region of the ligule. It's a process really reminiscent to what you would see at the back of a CD. Nobody look at CD anymore, but if you find one, look at the back. And this is the same process used to create color from perfectly transparent material. And during my postdoc in the lab of Beverly Glover, we showed that this is actually quite a widespread trick that are used by a range of flowering plant species across the phylogenetic tree of Angiosperm. And this semi-ordered striation can produce this specific type of iridescence that shifted toward the blue UV end of the spectrum. And this signal can increase flower salience and bee foraging. But the next question is really, how can epidermis cells control what is happening on their surface? And to try to answer this question, I started during my postdoc to develop a new model system. So this is a species of hibiscus, but it's not like the woody hibiscus you will find in park or garden center. It's really a weed. It's like the Arabidopsis of hibiscus. And it ticks all sorts of boxes of a good model system. It's very hard to kill. It grows easily. And we've now developed a range of resources and tools that facilitate experimentation. So like the daisy I showed you earlier, this hibiscus possesses very heavily striation of the cuticle in the basal portion of its petal, and that creates this blue metallic sheen you can see. And to cut a long story short, what we found recently is that by combining mathematical modeling with chemical analysis and mechanical perturbation, it's really the composition of the cuticle and chemical composition of this cuticle that play a crucial role in restricting the formation of the striation to the base of the petal, so that we were able to induce the formation of this striation on the surface of petals that have not striated yet, simply by stretching this petal and applying a mechanical force to them. This is a work we've done in collaboration with Sarah Robinson. But we can only do that if the cuticle chemistry is right. If we stretch petal from transgenic light that have exhibit some modification of the cuticle chemistry, the striations fail to form. So this striation, they are one part of a bigger pattern, because next to the region that contain the pigment and the striation, we find a rest of the petal that's very different. So in the top part of the petal, the cells are white, they are conical and not flat and elongated like the cell we found at the base of the petal. And they're completely smooth, there is no striation there. So this is very interesting because it shows that if you want to create this bullseye pattern, during the development of the petal, there must be mechanism at play to design a line across the epidermis of the petal, allowing cell on either side of this boundary to develop very distinct characteristics, different ability to produce pigment, different texture, and different cell shape. And the question is really, how is this boundary laid down? So this boundary, we can actually see it. If you zoom at the interface between the two regions in mature flower, you find cells that are flat and elongated, like the cell we've got at the base of the petal, but they are completely smooth. So they have the shape of the cell of the proximal region and the texture of the cell in the top region. So to try to understand how the different elements of this bullseye came about during development, we looked at dissected petal, and what we found is that actually the first sign of bullseye patterning appeared at what we call stage 2 of hibiscus flower development. And at that stage, it's only a difference in pigmentation. The difference in cell shape and texture come much later during development. And at stage 1, the petal appear completely unpatterned. So we did different things to try to find out which genes were involved in these differences. And one approach we use is a comparative transcriptomic approach. And the idea there is that some of the genes that must be involved in laying down the foundation of the bullseye boundary must be differently expressed between the two regions. Not all of them, but some should be. So we dissected petal just before bullseye emergence and just after, separating the different regions of the petal, and extracted RNA and produced transcriptome. And what we found is that even at stage 1, when the petal look completely unpatterned, the two regions of the petal already express very different sets of genes. So this means that the cells are already engaged in very different developmental trajectory. So we selected some of these genes, and we use our transformation protocol to modify their expression in planta. So this is a regeneration protocol that allows us to recover effectively transgenic line, or genome-edited line as well with CRISPR. So from the work we had done on striation formation, we knew it was possible to only modify presence-absence of striation without modifying the rest of the bullseye. And what we found by selecting this gene from our transcriptomic study is we could also do the same with the other features. So we got line in which the pigmentation is modified, line in which cell shape is modified. For instance, here, the conical cell can be flattened, replaced by flat elongated cell, or even replaced by jigsaw puzzle cell. In some cases, we got all the elements of the bullseye correct, but their distribution is modified so that the proportion of the whole pattern are changed, and the bullseye is either larger or smaller. And finally, we got line in which the bullseye boundary formation is impaired. So instead of having a neat boundary, you have now this really diffuse transition between the two regions. So it's very nice to find the gene, to find the gene that are involved in petal patterning. And now we have lots of work left to do to investigate these genes, understand their mode of action, and really reconstruct the gene regulatory network that allow the flower to control when, where, and how to lay down boundary in the petal epidermis. But this is only one part of the story. One thing we really want to do is to link gene activity to cellular behavior. And to do that, we've generated some reporter lines. So a line, for instance, that allow us to track cell polarity or the behavior of the cytoskeleton. And the idea is we are using this line now to understand how change in gene activity translate in different cell behavior and the emergence of different cell shape as the bullseye emerge. And we also want to understand how this cell behavior translates at the bigger scale to explain the overall organization of the bullseye. What do I mean by that? So you can imagine that we found actually there is probably more than one mechanism to generate petal with various dimensions. For instance, let's imagine that you have a line of cells that go from the base of the petal to the tip. To specify the proportion of the bullseye, you need to do two things. You need to specify the boundary during development, the position of the boundary, so that the cells that are below this boundary will develop the characteristic of the basal cell, and the cells that are above this boundary will differentiate it to top cell. We can see two different ways of generating the bigger bullseye. Either you can modify patterning mechanism. So now the boundary is specified higher up along the base to tip axis of the petal. Or you can maintain patterning mechanism as in the reference line, but instead you change growth either side of the boundary, either by modifying cell expansion or cell proliferation. For instance, if you reduce growth in the top and expand growth in the bottom, you can generate a bigger bullseye. So to try to understand what process was at play when we generated flower with a bigger bullseye, we thought we need to do two things. We need to be able to track boundary specification, and we need to be able to track growth. And Lucy in our lab developed a confocal imaging pipeline to do just that. So she imaged the full surface of the petal. I'm only showing you one stripe. But the idea is she can track cell behavior. And what she found is that the bigger cells that are here colored in red were not found at the base or at the tip of the petal like we expected, but instead were found at the 1/3 position from the base to tip axis of the petal. And this is really the position where the future bullseye boundary will form. So here it's a better way of representing the data. On the x-axis, you've got the relative position of the cell, base of the petal tip, and their size along the y-axis. And you see that there is a maximum of size around the 1/3 position. And what's really interesting is actually Lucy had to go back quite a long way back in time during development to find a petal that failed to show this behavior. So what does it tell us? It tells us that the layout of the bullseye is set up long before the bullseye emerged. So the petal is really pre-patterned. And we are now developing genetic marker to track identity establishment of the different compartments. So we've got report of a distal and proximal region, and we are in the process of generating this boundary marker. And this will be really useful for us to understand what is really happening when we obtain mutant or line in which boundary formation and petal patterning is severely disrupted. Okay, so the second thing that we needed to track is growth. So Lucy is tracking cell expansion, but she also found a way of labeling cell division event. So she could really characterize cell proliferation spatially. And this is what she found in the wild type, where most cell division events are restricted to the top of the petal. This is really interesting because it really provides us with a way to investigate what is happening in transgenic line with variation in bullseye dimension. So we obtain this line with a bigger bullseye when we constitutively overexpress a transcription factor from the TCP family. And what we found is that in this line, the pre-pattern is unaffected. The bullseye boundary is specified at the same position as in wild type. But cell proliferation is increased at the base of the petal. And this is what we think creates the increase in size of the bullseye. So what we are doing now is really taking full advantage of the presence of our colleague at SLCU, which are experts in computational simulation. And we are combining this simulation with our experimental data to try to understand, to really test model that would explain both the establishment of the bullseye boundary and its maintenance during petal development, both from a developmental viewpoint, but also from an evolutionary viewpoint, and how the network that control this process can emerge during evolution. The million-dollar question, of course, for us is what are the signaling processes that are laying down the foundation of the pre-pattern? And we are looking at obvious culprits like auxin, for instance, taking advantage of our reporter line that allows us to track auxin response dynamic during development, but also very interesting in other small molecules, because when we disrupt the production of this small molecule, we observe very interesting and striking defect, both in petal organogenesis, but also in petal patterning. Okay, so of course, nature did not wait for us to create mutant or transgenic line to generate diversity. And our little model species, Hibiscus trionum, belong to a bigger family known as the trionum complex. And this group of plants is widespread in Australia and in New Zealand. And it's known as the trianom complex because until recently, we didn't really know the relationship between them. And this group is interested for us because similar to what Yao explained with his really nice Mimulus work, there is some interesting pattern variation between species, but also between population of the same species. So for instance, there is change in pigmentation, but also loss of the bullseye or reduction in bullseye size. In some accession, we even just, it's the boundary that stands out because that's the only part of the petal that seems to retain pigmentation. So to try to understand how the bullseye has evolved during speciation and the radiation of this group of plants, we needed to have an idea of the relationship between species. But when we looked at classic marker used to solve relationship between species, we did not get any information because it's probably a case of recent radiation. They were really, really similar, near identical to each other. So we had to come up with another strategy, and we use a genome-wide approach, so phylogenomic tools that allow us to extract meaning from the noise of the evolution of entire gene family. So we generated transcriptome for this accession, and instead of just generating a handful of gene tree, we reconstructed gene tree for entire thousands of orthogroups. And this really helped us figuring out how these different species and population were related with each other. I'm only showing you a cartoon depiction of the relationship. And now that we've got this relationship, we can investigate the evolution of the bullseye. So what we found is, for instance, there is at least 4 independent events where the bullseye was lost or reduced during the evolution of the group. And to try to understand the genetic underpinning of this transition, we decided to focus on the sister species of our model species, Hibiscus richardsonii. So the pigmentation is clearly reduced, and May and Alice were able to show this. This is a Mendelian trait. And we, to cut a very long story short, we were able to find that this reduction in bullseye is due to a modification of a small gene regulatory network that account for pigment production. So in trianom, we found that the production of pigment at the base of the petal is regulated by 3 transcription factors that restrict the expression of DFR1, the enzyme that produces the pigment, to the petal base. One of these transcription factor, CREAM, is a repressor because if we force its expression everywhere, we generate flower with paler bullseye. The other 2 are activator because they have the opposite effect. When we express them everywhere, we create ectopic pigmentation in the top part of the petal. And at first, we thought that maybe the reduction in bullseye size in richardsonii was due to an increase in the activity of the repressor. This did not turn out to be the case. Instead, the bullseye reduction is due to a loss of function allele of BERI1. And this allele is expressed at very, very low level and contains some premature stop codon. So even if protein is produced, it's probably non-functional. Interestingly, what we found is that in 2 different populations of a different species of the complex, the bullseye loss correlates with the loss of the same gene. The homologue of BERI1 in this population seems to have been completely lost from the genome. Of course, we can now investigate the evolution of the other features of the bullseye and other traits. And Erin joined our lab recently to combine population genetic and field work to try to do just that. Okay, finally, what do pollinators think about this change of bullseye dimension? The first question we wanted to ask bumblebees, can you see a difference when you have 2 bullseye, but they only differ based on their size? So we did something very simple. To avoid bringing confounding factor, we just started with 3D printed discs that differ only in the size of their central bullseye. And we could tell the difference between them very, very easily. And they even show some clear preferences for the disc with a bigger bullseye when both discs were equally rewarding. And this preference was actually even stronger when we repeated this experiment with real flower. So what we are trying to do now is we're really trying to exploit this collection of transgenic and mutant lines we have that show more subtle variation in bullseye size. And this will help us understand what is the resolution limit for pollinator. When the 2 bullseye are different enough in size that they can be perceived as different and maybe contribute to speciation. And we can also, of course, start to really disentangle the contribution of the chemical element, the pigment, and the structural element, cell shape and structure. For example, in the line you've got on this side, the DFR1 enzyme that makes the pigment has been completely silent. So there is no anthocyanin pigment, but we still see a bullseye because the texture and the cell shape are different. And the question is, can bumblebee and other pollinator really perceive this pattern as a bullseye as well? So what I try to do today is to give you a small overview of the multi-scale and interdisciplinary strategy we're using to answer what we think is a really fundamental question that's central to our understanding of our life work. How can decision made by cell using molecular process can have an impact at the macro scale and influence organism fitness and the production of biodiversity? So of course, beside the sheer joy of kind of exploring how living organisms are wired, this can also lead to very practical application, because we know that 90% of flowering plant species rely on some form of plant-pollinator, plant-animal interaction for their pollination, and about a third of what we also rely on this interaction. So by trying to understand how a plant can engineer features to communicate with their pollinator, we hope we can contribute somehow to providing the knowledge required for food security, but also to promote the conservation of biodiversity. So to finish, I want to thank the current and past members of the lab that were really courageous people to jump in the boat of a new PI on a new model system. I only had time to show a glimpse of everyone's work today, but science is teamwork, and it was important for me to really broadcast how these people work together. I also want to thank the support staff we've got at SLCU. We are really lucky. We have fantastic support provided to us on a daily basis, and a mentor and collaborator either in Cambridge or elsewhere. Of course, the founder that allow us to do this work. And finally, thanking everyone for your attention and listening. Thank you.
Thank you, Dr. Moirod, for the beautiful talk. And now it's our Q&A section. Any questions from our committee members?
All right, I'll start again then. I was just wondering, because of this preference of the bumblebee for the bullseye size, whether the bullseye correlates with some other feature like nectar production or so, because the cells are also different.
Yeah, that's a great question. So that's something we are looking at the moment. We seem to have variation in nectar production, for instance. We don't know yet if there is correlation or not, but we're investigating. But there is definitely correlation with other traits. Like we noticed that when the pigmentation recedes on the petal, it also recedes on the ovary. So there is sometimes correlation because things might be coupled developmentally, but there might also be correlation because the size of the bullseye is indicative of the amount of reward, for instance. Maybe the amount of pollen, or it could be the amount of nectar, but we don't know yet.
Thank you.
I had a question on the boundary specification. So if you look to the distribution of cell sizes, you showed that actually you have small cells in the distal side, and then in the center you have big cell size, whereas when you go to the proximal side, you again have smaller cells.
Yeah,
this is correct. Yeah. So
yeah.
My naive idea, because I'm really trained by Arabidopsis research and so on leaf development. There you have this what is described for a leaf that is developing, that you have a kind of wave of cell division that goes from the distal side to the proximal side, followed by a wave of cell expansion, eventually combined with endoreduplication and so. But in any case, in this leaf structure, you have a kind of situation, I guess, where you don't have actually larger cells in the center. Or how do we have to interpret this as at the level of a kind of kinematic analysis? Do we have cell proliferation activity at both sides, proximal and distal side, or how does this work? And normally you have a gradient from proximal to from apical to basal side is a kind of gradient, but here apparently not.
Yeah, so that's exactly what we expected. And very early on, at the beginning of primordial emergence, we can see what would be like a gradient of cell division. There is always more cell division at the top. But what's clear is that the cell division stopped very, very early on at the base, much earlier than at the top. It carry on at the top almost all the way to flowering. So our current idea that actually what specify the boundary is an external signal that instructs the cell to stop dividing and start expanding. So we need to test this hypothesis, but we think that the cells are bigger because they're the first one that stop dividing and start expanding.
Right, but then at the very proximal site, you still have smaller cells.
Yeah.
And how they can be smaller? That means that they probably divided longer or still?
Either they divided longer, either we think there might be a gradient of cell differentiation from the boundary going down. So an opposite gradient that would go, the first cell start, so cell elongation is delayed. Because at the end, the cell size is homogeneous across the proximal region. So the cell all end up being the same length. But the cell at the base start elongating after the cell at the boundary. Okay, if that makes sense.
Okay, yeah, thank you.
You're welcome.
Okay, let's talk. So I have a general question. So have you identified any key molecular or hormonal pathways determine the petal pattern? Except you talk about more about the Book's boundary. So I mean, have you identified any key pathways determine like the surface structure, petal shape that are different from the pathways in Arabidopsis or in monkey flower, just like the Yao taught.
So two things, if I understand the question properly. So we look at obvious pathway like auxin, and long story short, we think auxin is very important to control the dimension of the bullseye, but not to set the boundary. Mm-hmm. To set the boundary, we have data suggesting that some small molecules are involved. And this is not necessarily something that is known in other species, but it does not mean that they are not doing the same thing in other species. It might just be that we have a striking pattern in a flower that gives us easy access to very early stage of development that we could not access easily in Arabidopsis, for instance. So, of course, the interesting question is we're not just trying to find out what happened in a hibiscus. We're using it to really try to understand fundamental process. And there is also a very good chance that what is used to pattern the petal from base to tip, it's also involved in patterning the leaf, especially from an evolutionary viewpoint. Flower, petal are modified leaves. So what we think we're doing is we are finding new things by using a different system. But these processes and signaling process might as well be at play in other systems. And that would be that's why it's nice to have so many groups working on different systems to see what is universal and what is a peculiarity. Does that answer your question?
Okay, thank you. Thank you.
Okay.